Hello everyone, so my name is Raphael Meudec. I'm a PhD student working with Bertrand Thirion and Damian Wasserman. Today I'm going to talk about our work that has been recently published in Nature Scientific Reports, which tackles decoding statistical maps at scale by leveraging open data initiatives such as NeuroVolt or Cognitive Atlas. So if we start with a quick refresher on fMRI data, we record the bold signal while the patients receive a specific stimuli or perform certain tasks. And we compare the signal acquisition to a contrast and deduce from there a statistical map where we can identify regions that are more specific to the given stimuli. And we have an interest in two tasks from there: encoding, which is inferring the regions of the brain implicated in a given cognitive process, and decoding, which is infer which cognitive processes are implicated for a given statistical map. So first, this work couldn't have been done without NeuroVolt, which is an open database of task fMRI data, which was built by Chris Gorgolewski and the Poldrack Lab. So it contains a large number of images, which are associated with some labels and some description. And it's an open data initiative, so everyone can update elements to NeuroVolt to visualize them in the browser or download and upload new elements through the API. So this allowed us to have access to thousands of fMRI images. Another open data initiative which has been useful to this work is Cognitive Atlas, which is a collaborative project to create a knowledge base of cognitive neuroscience. It insists on the separation between concepts, cognitive processes, and the tasks that allow to measure them, and it organizes them in a hierarchy. So we focus on the decoding setting, which is a multi-label classification setting. So for a given map, we aim at predicting a list of tags that has been assigned to the map by users in NeuroVolt. And the pipeline is divided in two parts. One is regarding the handling of the labels, and the other one regarding the images. So on the images, we first perform a map selection of Z or T maps. Also, we filter out elements which are empty images or which contain none values, which reduce the number of maps from NeuroVolt from 140,000 to around 50,000 images. We then preprocess them by resampling and masking and embed them into DFUMO, which is a dimensionality reduction technique built for fMRI data. We use a multi-layer perceptron to perform this multi-label task, a simple model with a hidden layer of 300 nodes and some regularization and dropouts, which outputs a multi-label prediction. It gives successful results of around 35 concepts, and we proceed in a left-out collection way. So here is the left-out collections is IBC, but we perform a cross-validation with other left-out collections. We also did some preprocessing on the label side by leveraging Cognitive Atlas. So some concepts imply other ones by their nature. For example, speech perceptions implies voice perception. So we use the Cognitive Atlas ontology to perform this hierarchy and add synonyms and hypernyms and enrich the initial vocabulary to a list of around 50 concepts to decode. It yields similar satisfying results, proving that we managed to do decoding at scale in neuroimagery. An additional interesting result is that nonlinear model we use yields better performance than linear models, which was not necessarily the case in the past. And this performance boost might come from the volume of data available, which is becoming quite significant. Another interesting fact is that some concepts are highly available in the training set, but their performance is not convincing. And this can be due to ill-defined concepts which regroup too much different tasks. We also looked at the sensitivity maps we obtained, which we obtained by differentiation of the outputs over the inputs. It shows plausible maps for well-decoded elements, such as left finger response execution. And on the contrary, terms that have an AUC of just above 0.5 do not capture relevant regions. We also see some other interesting effects on the sensitivity maps. So, for example, TOE response execution is a well-known task. Decoding maps are properly identified. For syntax, it focuses more on Broca's area, which is consistent with the literature. For emotion perception, we see that the faces area is still present and may be the tradition of a bias in the experiment where the emotion perception task is usually performed on faces. So, as a conclusion, we leverage NeuroVault data and cognitive atlas initiatives to scale the volume of data to around 50,000 maps, and we successfully decode at scale those fMRI maps on around 50 different concepts.